Okay, is it good? And you can see my slides?
Yes.
All right, good. Then the key things are in place. Well, I'm so glad that everybody is able to come and join us. Thank you, Shane, for the introduction. My name is Dr. Matthews, and I am a new faculty member here with the School of Mines in Rapid City. I'm in the Department of Chemistry, Biology, and Health Sciences. Previously, I was with the University of Texas system for 5 years before joining up here, and my background is in infectious disease epidemiology, which makes me very much in demand right now. My husband is particularly happy that all my years of graduate school are paying off at this point in time, so he's enjoying that. Just a little bit about my background. So I have a PhD in epidemiology. Specialization is in infectious diseases. I teach courses in immunology, microbiology, genetics, physiology, and also epidemiology. So I have kind of a broad range of topics that I cover. Basically anything to do with infectious diseases is really what I'm passionate about. But I also have a master's degree in public health. So I have an interest in not just understanding some of the technical side of how things work in like a laboratory setting if you were doing research, but also what the applications are to kind of the big picture. How do we bring this back to concepts of public health in our communities, in our nation, in our world, and so forth. As was mentioned by Shane, I do have a close tie to the School of Mines. A lot of my family members have gone here, which is really fun for me because I did not go here and now I get to teach here. So I'm delighted to be here with the department. Now, in terms of the particular talk that I picked, I decided to choose this topic focusing on comparing the SARS coronavirus that is currently in circulation with influenza. And the reason why I picked these two viruses is because I have heard a lot of questions, I have a lot of comments, I have seen a lot of things on social media, I have seen a lot of things on news sources and magazines, all looking at trying to compare and understand and conceptualize the similarities and differences between these two viruses. I have heard a lot of statements of things like, this is just a flu. It's no big deal. The flu kills lots of people. It's a respiratory illness like the flu. You know, when flu season comes, we'll get flu shots or we can take drugs and it will be okay and it won't be a problem. So I've heard a lot of that. I've had a lot of questions about that. I've had a lot of people say that they don't understand if coronavirus is a respiratory illness, why you see some of the symptoms you do, especially if it's not like influenza. A lot of comparisons are made to things like the 1918, big global pandemic, which was an influenza pandemic. And then there's also a lot of questions and concerns about vaccines. I've heard criticisms and comments that say things like, well, if we can't even figure out how to get a good flu vaccine, what makes us think that we can figure out a COVID vaccine? I have heard people ask questions about, whether SARS is going to have seasonal transmission like we see with flu, if it's going to be mutating the way we see and have issues with flu. So I thought, in light of all of the information, because there is so much information that is out there, and trying to understand some of these questions and follow along with these topics that we see, especially in the news media and what we call secondary sources, I thought it might be really helpful to go back to some of the basic biology and really kind of dive in and do a comparison between these viruses. So here is the agenda for today and what I want to cover in this talk. First of all, I want to go through and do a comparison of basic viral structures. So this is where we get to do a little bit of basic virology. We get to cover some topics and what viruses are and what their physical structures are. And then from there, we're going to lead into kind of this discussion on viral genetics and replication, which is really providing a lot of the framework for understanding what is taking place when we are having an infection, when viruses are coming into a host cell, when they're replicating in a host cell, and then when they're also leaving, because all of those steps are important for understanding the dynamics of how a disease works. And then after we kind of lay the groundwork through all of that, we're going to bring that back to talk about how each of these components have applications to the current vaccine development. Conveniently, both Moderna and Pfizer have come out with information in the past couple of weeks talking about the efficacy of some of their vaccines. There's a lot of excitement about the potential for a vaccine coming up. We attended a talk with our state health department for South Dakota on Tuesday where they're discussing all of their plans and trying to get everything ready to go. So in light of the fact that vaccines are now in the forefront of kind of what everybody's thinking about, I thought it would be really good to kind of look at and understand how we're designing these vaccines. What are some of the difficulties for both influenza and coronavirus that we're having with them, and maybe for COVID, some of the things we can expect as we're coming forward in the next year. So let's start by talking a little bit about some basic virology. Let's understand a little bit about what viruses are and what are the main components that are in here. So for those of you that, remember back to your days, I don't know how familiar your biology is or how long it's been since you've done any biology. Some of you, it may be right in the forefront of your mind, and others you may have to rifle through that memory cabinet a little bit. So we're going to be pulling out some of those basic biology topics. If you remember going through grade school and learning about the structure of a cell, you typically do that project of like a diorama where you make a cell where you have the nucleus and you have the organelles. Most people joke that they get out of high school remembering that the mitochondria is the powerhouse of the cell, but they don't remember much else. So all of those things that you learned kind of about those cells that you might remember, kind of hold on to that, but also kind of set it aside, because a lot of those things are not really relevant when we're talking about viruses. So some words that you might remember learning about genetic material that cells carry. So a virus fundamentally has to have some kind of genetic material, just like you would see in other cells. Most people remember DNA and are familiar with it. So DNA would be your double helix, where you have your base pairs that come together and it forms your genetic code that creates the instruction manual for your cell, for your body, and everything else. So viruses all contain genetic material, but they're a little unique in the fact that they don't always have DNA as their genetic material. If you remember this structure called RNA, RNA is a single-stranded nucleotide that's made from DNA. You basically take DNA and you make a copy of it, and then you create a single strand, and that's your RNA. So viruses can contain genetic material in their very center, and they can have DNA and RNA. And then importantly, they are coded by some sort of what's called a protein capsid. So when we talk about proteins in biology, we're not really referring to nutritional proteins. We're talking about when you take amino acids. And if you remember, when you have the structure of protein, you kind of string amino acids together, kind of like you were making a necklace with beads. And then you take that necklace and fold it into a three-dimensional pattern. And when you get that three-dimensional shape, that represents a protein. So viruses all contain genetic information wrapped up in some sort of a protein coat. And this capsid can have cool shapes, kind of like icosahedral shapes that you might see, or it can be a capsid that is very close to the nuclear material and actually kind of wraps around it almost like a little envelope or sheath. So basically, fundamentally, this is what a virus is. So it's much more simple than what we see in other more complicated cells like human cells, plant cells, even bacteria are much more complicated than this. There's no organelles in here, there's no mitochondria in here, there's no nucleus in here, none of that exists. The virus is just genetic material wrapped up in protein. Some viruses have an optional what's called lipid envelope. Now this is something not all viruses have. If it's missing it, it's called a naked virus. But what's important about this is that enveloped viruses have slightly different transmission dynamics than non-enveloped viruses. Importantly, viruses that have an envelope tend to be more susceptible to environmental conditions, and so they don't tend to survive on surfaces as long. So like for coronavirus, when they say, it doesn't survive on surfaces more than like a maximum of 3 to 4 days, the reason is because it's a lipid-coated virus, and so it becomes inactivated over time. Now I use this word inactivated because there's two other components that go along to defining viruses. Viruses are arguably considered to be what we call obligate parasites. Viruses are not capable of surviving in and on their own. But importantly, the second part that goes along with this is that viruses are classified as being non-living. And some people have a hard time wrapping their head around this conceptually because we talk about killing viruses and killing them because they cause disease. When we talk about things that are living organisms in biology, like I teach microbiology, and so when we're in microbiology, we have to actually grow our bacteria, we have to give them a food source, we have to put them in a little warm incubator, we have to make them happy, we have to nutritionally give them everything they need in order to go through all their biological processes in order to grow and replicate. Viruses don't do any of that. The entire job of a virus is to take this genetic material and find a way to copy it and then repackage it and send it on to the next host. So because of this, I like to refer to viruses a little more casually sometimes as the ninja pirates of the biological world, because what they have to do is they have to find a way to sneak into a host cell without tripping any alarms or activating any of the immunological surveillance systems. And then on top of that, they have to hijack the machinery that's inside a host cell, and they use that host cell machinery to make their copies and then transmit themselves. So all of these things are very key to understanding how viruses are causing infections and how transmission dynamics work. So let's look a little bit at the structure of influenza first. This is a little figure that represents a cartoon graph of what influenza looks like. So some important key things that you need to know from this. First, influenza actually has 8 different segments of genetic material. If you think about genetic segments, kind of like we have chromosomes in our cell, influenza has 8 single segments of what's called single-stranded RNA. So it's an RNA virus. It has a viral capsid that is wrapped around the nuclear material, and it does have this lipid envelope around the outside of it. Now, the 2 main proteins that we're going to be talking about are surface proteins that allow for the virus to enter into a host cell and then also leave. So you've probably heard about hemagglutinin at some point in time. It's abbreviated as HA. Hemagglutinin is this little blue one right here, and it represents a surface protein that allows for a virus to have entry into a host cell. Think of this as the equivalent of like having the right passcode to get in through a door. You have to have the proper communication for a cell to allow the virus to come in. And so the hemagglutinins interaction with the surface of a host cell is what's going to provide that signal to allow the virus to come into the cell. Now, influenza also has a protein that it needs for exiting a host cell properly in order to be transmitted. And this is what's called neuraminidase, and it's abbreviated as an NA. Now, there's other proteins that are in here, but these are the key players that we are going to be focusing on for today. Now, influenza has 3 main different types that we classify it as. We have influenza A, B, C, and then technically there's a D that's not able to infect humans. Influenza C arguably doesn't have a true hemagglutinin or neuraminidase, and it typically doesn't cause very significant clinical disease. The big ones that we talk about are influenza A and influenza B, and those are the ones that you find in the seasonal vaccine. Now you'll notice for influenza B, we just have 2 different versions of it. These are different lineage strains for the virus. So the main one we typically focus on is influenza A, and this is abbreviated as an IVA, influenza virus A. So what's important about influenza A is if you notice these H's and these N's right here, every one of these numbers represents a slightly different version of each of these surface proteins. And the versions of these surface proteins are going to dictate how well a virus is binding to a host cell and getting inside, how well it's getting out of a host cell and being transmitted, and is really key in understanding some of our pathogenesis and our transmission dynamics. The other thing that's important about influenza A is it's the only version that can be transmitted between both human and non-human hosts back and forth. So for things like birds and pigs and humans, and we'll talk about why that's important a little bit later in the presentation, but that's an important point of influenza A. Now, a lot of our seasonal strains of flu that we see are going to be influenza A. So when we write out like our H3N2, when we put this in, this is representing the different versions of hemagglutinin and neuraminidase that are present. So our typical circulating strains are going to be H3N2s and H1N1s. H1N1 versions also tend to have more pandemic potential, and H1N1 is believed to have been the 1918 version of an influenza and was also the type we saw with the 2009 swine flu outbreak that was happening. So let's contrast this now with coronaviruses and look at some of the differences and similarities. So right off the bat, they look pretty similar. A lot of people see pictures of them and they look like spiky balls. And so they look very, very close together. But the biggest difference that you'll see right from the beginning is that coronavirus has a different structure for its genetic material. Now it's also an RNA virus, so it carries RNA as its genetic material, but it doesn't have these 8 individual strands. It has one long genome, and in fact, it has one of the longest genomes in the RNA virus family. It also has a viral capsid, which is referred to as the nucleoprotein, and this is important for some of the vaccine candidates a little bit later, and it's also a lipid virus, so it contains this lipid envelope. Our main surface protein, though, is not going to be hemagglutinin or neuraminidase. Instead, we have a different structural protein that's known as the spike protein. It's abbreviated with an S. This spike protein is the key protein that allows for viral entry into a host cell, and it is the major player that we are looking at when we are trying to develop vaccines. So these are the major players for coronavirus. Now, this right here is a graphical representation of the genetic similarities between different families of coronaviruses. So a couple of things I want to point out here. First of all, we already have coronaviruses that circulate in human populations that cause common colds. These are our 4 main strains. You can see coronaviruses are divided into alpha coronaviruses up here at the top. We have our beta coronaviruses over here, delta coronaviruses, and gamma coronaviruses. So anything that has an H in the front of it represents something that infects human populations. So these are already in circulation. We already have common colds that occur because of them. But these aren't the ones that we really worry about in terms of, really serious disease. Those would be these guys right here. This one up here is the MERS coronavirus, the Middle Eastern Respiratory Syndrome. This is a version of coronavirus that very fortunately so far has not shown good human-to-human transmission. So we've had some small outbreaks where we have individuals or small clusters of cases, but we don't see widespread transmission, which is good because right now the fatality rate is sitting somewhere at about 34% for this particular coronavirus. So one of the big concerns is that we want to make sure that this one is not being able to develop the ability to be transmitted easily between humans, because that could be very problematic. The other one is this down here, which is the SARS. And the SARS, of course, stands for Severe Acute Respiratory Syndrome. So this was the original SARS coronavirus. Our second SARS coronavirus is very similar in here. They have what's called an 80% sequence homology. This just means that their genetic sequences are about 80% similar. And they're very similar, especially in some of the key components, things like the spike protein and other things like that. Now, what you also might notice is that for MERS and SARS, they are clustered together with all of these bats, these bat coronaviruses, and this is no surprise. Bats are one of the primary, what we call reservoir hosts for coronaviruses. They circulate very heavily in bat populations, particularly in horseshoe bats, which geographically tend to be found extensively throughout parts of China and Southeast Asia. This is why it is absolutely no surprise that for the last 2 coronavirus outbreaks from SARS specifically, that they have been geographically located in this particular region of the world. And so you've had researchers over in parts of China and Southeastern Asia following and tracking bat populations and coronaviruses, especially for the last 10 plus years since the last SARS outbreak, because they know that this is something that's very important. All right, so that's just a very brief overview of some of the backgrounds of the viruses. So let's switch gears a little bit and talk a little bit about viral genetics and replication cycles for these viruses. We have to make sure that we understand this, what's called the central dogma of molecular genetics and biology first, because this is really foundational and key for understanding this whole next section. So as a quick reminder, and this is where you can dust off some of that biology from a little while ago. So here is our DNA. It is a double-stranded helix. In genetics, in biology, our key central dogma is that their DNA that codes for genetic information has to be translated into some sort of a product. And in this case, we talk about the production of proteins, but there has to be an intermediate step. From DNA, we have to transcribe it into what's called a messenger RNA, or a single-stranded RNA. And then importantly, this RNA template serves as a template to be able to translate using machinery in cells. And if you remember learning about ribosomes, ribosomes are the machinery that are translating RNA into proteins. Now, proteins are very important. If you remember, so here is a structure of a protein. So this would represent your single chain of amino acids. So starting from your RNA, each one of these amino acids are coded. So we have like a genetic code that we have to follow where we're programming and creating these amino acids. What's neat about these amino acids is each one of them has slightly different chemical properties based on their structures. So when you put them all together and then they start interacting using chemistry, they begin to fold up on each other into a 3-dimensional shape that looks like this. So you'll see these proteins illustrated sometimes with these wavy, curvy lines, and this represents this single chain that has been folded up in a pattern based on the chemistry. And this is really, really important because this 3-dimensional folded shape is absolutely key for understanding how proteins are interacting, and really importantly as well, changes to your messenger RNA template that can affect this amino acid sequence can have an impact on how you're doing your 3-dimensional folding and how proteins are going to be interacting in an environment. So imagine if you have this entire sequence of these circular beads, say you have a mutation that occurs in your RNA, you can have a couple of different outcomes, but let's say theoretically it changes this little bead right here to a triangle or a star shape. just for kind of visual ideas. So if you go to fold this, if you have something that's structurally different here, it's going to affect the way that this is going to fold, and it may only affect a small little piece, but sometimes that tiny little piece makes all the difference, especially for the way that a virus can interact with a host cell. So we literally have to come down to look at changes sometimes to a single nucleotide and the effect it can have on amino acids and on this folding structure. So with that kind of framework in mind, let's talk a little bit about the genetics here in the viruses. This figure right here represents what we call the Baltimore system of classification of viruses. Our viruses are broken up based on their genetic material. You see you have DNA viruses on the left, RNA viruses in the middle, and then our retroviruses on the right-hand side. So what we're going to be focusing on today is we are going to be in the RNA category. We are going to be looking at this group 4, which represents coronavirus, and then group 5, which represents influenza. Now you'll notice in here that you have RNA, but then it's got these pluses and minuses, and I'll explain that on the next slide. But what I want you to get out of this is that if you follow these groups down, you see that right here we have mRNA, and if you notice, everything on this figure is leading in towards the production of this messenger RNA. So no matter what the original genetic setup of the virus is, ultimately its goal is it has to produce this mRNA in a readable format so that it can be translated correctly into protein products. If this isn't here, you don't get these. And for viruses to replicate, they have to be able to produce proteins using the machinery that are in a host cell. So let's talk a little bit about what this plus and minus represent. The RNA plus represents a what's called positive sense RNA virus. The negative is a negative sense. So what does that mean? Let's start with an example with influenza. Influenza is a negative sense RNA virus, so it has a genome that looks like this. You cannot translate this directly into a protein, so you have to make a template off of this that is actually able to be translated. So a key for single-stranded RNA viruses that are negative sense is that they have to make a positive sense template first in order to produce proteins. So if you have, for example, on your negative strand, if you have these base pairs, now you might be looking at this and you're like, I don't remember you. I remember there being a T in there somewhere. Base pairs for DNA are A, T, C, and G, but for RNA, the T is switched out with uracil, so it's structurally very similar, but it technically is a U. But if you have, for example, this code right here, when you come to make your template, imagine that you're making a mirror image photocopy. So you're going to make what's called a complementary strand with the complement on here, and then you can use this strand to now serve as your template for creating proteins. So you're going to make a whole bunch of these and then use these to translate into your protein products. Additionally, you can use this strand, this positive one, now as a template for making more negative ones, because a virus not only has to make protein, but it also has to make more copies of its genome so it can put together little packages of new virions to send out. So the purpose of this template then is then to create and go back to making your original one, because again, you're making that mirror image photocopy, so it would go back to the original starting point. By contrast, a positive sense RNA virus is already starting with a usable template, so it's already in a correct format that it can be directly translated to proteins. Coronaviruses do something a little bit different. They throw some extra steps in there, and we'll show that in a minute, but technically you're starting with the correct template already. The important take-home point of this is that each of these steps represents an opportunity to introduce mutations into genetic material. Now, when we do this same process that's known as transcription and translation in our cells, we actually have proofreading mechanisms that go through and check for errors that may have occurred, and we have systems available at the DNA level to correct errors. Now, this is really important for preventing unwanted mutations that can occur every time you have to replicate a cell. For influenza, importantly, influenza does not possess the ability to do proofreading, which means that as it is replicating, which it tends to do in somewhat of an error-prone fashion, it introduces mutations without a system to be able to check and correct them. Coronaviruses do actually technically have some ability to do a little bit of proofreading. It's not the same as what you see in humans, but they do. So one of the things that people ask is, well, you know, influenza has a lot of mutations, COVID is coming out, are we going to be seeing mutations? That's part of what we're going to try to answer in this series right here. But to start with, to understand, influenza already is going to be more error prone simply because it has no proofreading mechanisms built in. Another key thing that goes along with understanding mutations, particularly with viruses, is this concept right here. This little graph represents our relationship between your genome size and then the mutation rate that's expressed as a mutation per specific site of a nucleotide per the number of replication cycles. Now, an important idea of this is that things that are very small, like viruses over here, not only do they have small genomes, but because they have small genomes, they can replicate very quickly. So viruses can go through massive amounts of replication as compared to the amount of replication something with a very large genome would do. So this means that from a statistical point of view, there's a greater chance that mutations are going to come up, particularly between the cycles, and especially with the total number of cycles that exist. But this also, on a small scale, also plays a role. So this is showing you everything from viruses up to something big like a mouse. Humans would be a little further out past a mouse. But just for some numbers to kind of make this have a little sense, the total size of the influenza genome is about 13.6 kilobases. The kilobase represents about 1,000 base pairs, and a base pair would be one of those letters like an A, a U, a G, a C. So each one of those is a base. So that means there's about 13,000 base pairs in an influenza genome. This is compared to the coronavirus, the SARS-2, which is currently circulating, which has a genome size of about 29.8 kilobases. So already it is, twice the size of influenza, so it's much bigger. And just for funsies, to kind of compare it to a human genome, the human genome is about 3.2 gigabase pairs, and a gigabase pair represents a billion. So we've got about 3 billion base pairs, so we are significantly larger. But this is another important concept in terms of understanding where mutations are coming from in viruses. Now, you might ask the question, what is the point of having mutations? Why is this something that viruses would want to do? The reason this is important for viruses is because mutations are considered to be drivers of what we refer to as fitness. Biological fitness represents how well an organism fits into its environment. So in the case here with a virus, its environment is going to be a host. So we're basically trying to understand how well is a virus interacting with its host, and can you improve the way that that interaction occurs? So since the interaction is fundamentally based on the interaction of a protein from a virus and an interaction with a receptor for a cell, that interaction right there, you can tweak based on having mutations that are going to change slight things in the proteins and its three-dimensional shape. Now, an important concept of mutations is that not all mutations are helpful. Some mutations are going to be problematic and actually prevent a virus from being able to do anything. It may actually destroy it. Some of them are completely neutral. The way that we do our coding in genetics means that sometimes you have changes that actually have no effect. And then sometimes they actually provide changes that give a little bit of a competitive edge to something like a virus. Now, importantly as well, when you have things that are newly emerging, for example, like the SARS coronavirus, it represents something that is relatively new in its host. So because of this, you would expect that it's going to be kind of testing things out and trying to get a slightly better fit. So as this has come out, people have been concerned, and there have been reports going around that say, oh, well, it's having mutations, it's changing, how much is it going to be changing? We would expect it to change. We would expect it to change because we would expect it to be trying to find a way to be better adapted to its host. And the longer it's going to be in a population, we would expect that to slowly over time kind of slow down a little bit and move to more like what we would see in example B right here, where mutations are still occurring, but you're going to have less of those beneficial ones because you're already pretty well adapted to your host. Now, for something that's been in a host for a very long time, it may actually reach a point where it no longer can find anything that's beneficial because it's already kind of at the peak of what it can do. Now, if you're following this logic, you may be asking, well, influenza's been around for a long time, so wouldn't you expect it to be well adapted to a host? So why is it continuing to have so many mutations? And that's a great question. So we have to come back and look at a particular concept that we discuss in influenza. So this is something we cover with our students. They absolutely love that these two vocabulary terms are one letter apart. It makes it really fun for learning for a test. But we have to talk about this idea of antigenic drift versus antigenic shift. So we're back to talking about influenza. Here are influenza viruses. We're actually going to start at the bottom here with this idea of antigenic drift. We've actually already covered this because what this represents is that if you have a genome that looks like this, and you have mutations that are occurring, mutations can be small changes. If you can see the small change, you have this little green piece right here. It is now corresponding to a slightly different hemagglutinin on the outside here. As you have influenza virus that's moving around, because it is an RNA virus and it tends to be error prone and it doesn't have proofreading capabilities, you tend to find these genetic changes that are going to provide slight variations within, see, we're still at H3N2. So even though it's technically the same virus, these slight variations are going to affect slightly how they're interacting with cells, how they're replicating, how they're being transmitted. And this gives rise to the variants we see in the different flu strains, where some years you have flus that are particularly awful and other years they're not so bad. A lot of that is mediated by some of this antigenic drift that's occurring as the virus is circulating around and traveling through the world. This is in contrast to what we call antigenic shift. This is something that influenza can do because influenza has these 8 individual pieces of genetic information. What is happening here is we have an example of an influenza that can infect a bird, but is not really infecting humans and causing a lot of disease that's widespread. And then we have a human virus that is able to infect humans well. Now, remember that I said that influenza A can infect and be transmitted between multiple species like birds and humans. What you can get in certain species is there is the potential that you can get a genetic mixing. So if you have a situation where a bird or a pig or a human is infected with 2 different versions of influenza that are capable of entering into a host cell and they get inside the host cell, imagine you are opening up and dumping out these 16 different pieces of genetic information, and now you're just going to kind of shake them up a little bit, and then when you go to repackage them, they get repackaged a little differently, so you might end up with a combination that looks like this. So now all of a sudden we have a new version of influenza that has the avian version of hemagglutinin and the human version of neuraminidase. This represents what we would call a potential pandemic situation where you have a pandemic outbreak of something that is new, that has occurred because of a significant genetic change that has occurred over a short period of time because of one of these genetic reassortment events. This is what scientists are worried about with what we call bird flu. So there are certain versions of hemagglutinin that are found in bird strains that we don't have in human strains. And there is a deep concern that there may be a genetic reassortment event that occurs that can produce one of these strains that is highly lethal and pathogenic in humans that then is able to effectively infect host cells and be transmitted. And this is why you see people responding to things like if they find certain strains of bird flu in birds, they go through and cull entire flocks, especially in parts of China and Southeast Asia, where we have seen some small sporadic cases of different types of bird flu getting into humans, but they have not been able to be transmitted yet. So the concern is that if there's enough genetic shift and change, that eventually that could arise and become another pandemic. And that's been something that people have been concerned about for a little while. Now, let's take all of these things we've been talking about and then bring them together and discuss how these are actually replicating in the host body. So here's influenza. For influenza to infect a cell, you have to have hemagglutinin here binding to a specific receptor. This receptor for influenza is what's known as sialic acid residue, and it's a particular form of sialic acid because there's lots of different ones in the body, but there's a particular structural one that's expressed on the airway epithelial cells, so your cells that line your respiratory system. So once you have this interaction right here, it's the equivalent of ringing a doorbell to let the cell know that you want to come in. If this connection is not proper right here, it would be like ringing a broken doorbell. The virus is going to sit out there and ring it, but nothing's going to happen. So when you have the proper interaction between the protein and the receptor, it's going to signal to the cell that you have the right password, you can come in. So you bring it in using a process called endocytosis, and then from here we have what's called fusion and uncoating. So you actually have this lipid part of the virus fusing with the lipid part of the cell, so they kind of meld together, and they just release all the genetic information out into the cell. And this is where it starts hijacking the machinery that's already available in the cell. So we're coming over here to the nucleus at the center of the host cell. Remember, we have to create that template of our RNA. So we're going to come in here and create that template. And from there, we're going to form this messenger RNA for making our proteins. And then we're also going to form our template for making new genetic material, which we're going to produce together right here. And then once we have all our components, we are then going to come out here, and for influenza, it likes to take all of its surface proteins and it sticks them in the top of the cell. And then it brings in the genetic information and it basically pinches off a piece of your own cell. So it's like your own cell is now moving around, but with the influenza proteins on the surface. And this is what's called budding. Now, there's something in here we haven't talked about yet, and that's neuraminidase. Here's what neuraminidase does. If you see in this illustration right here, as this is leaving, this right here looks exactly the same as this right here. And that's because as influenza is leaving, the hemagglutinin is actually going to bind back again to the sialic acid and basically get stuck like it's being tethered to the surface of the cell. So the job of neuraminidase is to act as a biological scissor and cut this and cleave it so that the virus can be released. So this is important because if neuraminidase is not structurally able to cut this tether, the virus cannot leave. And if it cannot leave, it cannot infect other cells, and it also can't be transmitted. This is the mechanism by which the major classes of flu drugs work. So like oseltamivir, which is Tamiflu, it is a neuraminidase inhibitor. So it blocks this mechanism right here so flu can't leave. But viruses love to genetically change and they love a challenge. So what we've basically done with Tamiflu is we've issued a genetic challenge to influenza to say, ha, we have countered you. So now influenza is going to sit in here, and as mutations are occurring, if a mutation occurs that changes neuraminidase in a way that this protein can no longer interact with this medication is no longer effective. And now you have a strain of influenza that is drug resistant, which is exactly what we see. We are seeing a rise of drug resistant strains of influenza that are in circulation because these ones are able to withstand and have countered our counter, so to speak. So now it's time for us to counter the counter to the counter. So we're kind of playing this game with viruses. This is the same mechanism that we see in antibiotic resistance, which in and of itself could be a whole nother talk. Let's compare this to coronavirus. So for coronavirus, when it gets into the cell, you have viral entry. Same thing here, except we are using a different receptor. You've probably heard about this ACE2 receptor. The interaction here, it's the same idea. You have to ring a doorbell to be able to be let in. The spike protein is binding to this ACE receptor. Coronavirus is coming in. What it does here, though, is a little funky. So remember I told you it's a positive sense RNA virus, so you would think it could go and directly make protein, but it decides to complicate things a little bit. It first creates a reverse template that it uses as another template to make its new strands of genetic information, but it also makes all these really short versions of messenger RNAs that code for each one of their specific proteins. So it's something a little unique in viruses, but the end idea is still the same. You still end up getting a replication of genetic material, you get production of proteins, and then what's also different about these is they don't actually pinch off pieces of the surface of the cell. They go over, if you remember from basic biology, your Golgi apparatus. It's an organelle in your cell. You can think of it as like being the FedEx of your cell. Its job is to do packaging and distribution. So it goes over and it actually pinches off pieces of the Golgi apparatus. It assembles itself there, and then it is released. Now, I have had people ask, and I've heard these comments about the use of Tamiflu for coronavirus. So as we just saw, the mechanism of Tamiflu is that it specifically interacts with neuraminidase, which coronavirus doesn't have. So you would not expect Tamiflu to be helpful for coronavirus. The class of drug that people are looking at, and you've probably heard of this one right here, which is Gilead's drug remdesivir. So the mechanism of action of remdesivir is that it comes in and it interacts with this protein that is doing the transcription so that you don't get copies of the virus. And that is how that works. Now, for ACE receptor, another thing that I've heard people make comments about is that, you know, compared to flu is a respiratory illness. Coronavirus seems to be causing symptoms everywhere. So it seems a little bit scammy, like they're just making up symptoms at this point in time. And I've had that expressed to me. The reason for this is because the receptor for influenza is found in the lungs specifically, but the ACE receptor is actually found in a lot of places in the body. It's heavily expressed in the GI system, which is why you get GI symptoms. It is only a little bit expressed in lungs, but it is upregulated, which means you have more of it for people who smoke, which is why smoking is a very strong risk factor for severe coronavirus. You also find it in the kidneys, you find it in the bloodstream. So all of these funky symptoms that we see can be traced back to the fact that the virus can actually go and interact with other organs. Same thing with the heart. You can have ACE that's expressed in the heart, and in patients that particularly are older and have heart failure, ACE can actually be increased so that you're more likely to have problems and complications. What's also interesting about ACE receptor is that there's a lot of comparisons to other mammals. So other mammals also have ACE, which is why we see the coronavirus in bats and a variety of other species. This is a fun little diagram that just kind of represents different mammal species that can or cannot bind with S protein. And just to give you kind of a little idea of some of the different species that are out there, bats, of course, are a big one that we already talked about. There's non-human primates like chimpanzees, baboons, orangutans. You have the pangolin and the palm civet, which have both been implicated as kind of intermediate hosts. Both of them have the ability to bind to the S protein. This one's been showing up in the news a lot, dogs and cats. Technically, yes, dogs and cats have the ability to interact with the S protein. However, dogs and cats are not the best host for it. And so you don't see a lot of viral replication, you don't see high viral loads, and there's not been any documented cases where it can be transmitted back from an animal to a human. But technically, yes, dogs and cats can be infected. Ferrets and stoats, hamsters, and then what I like to call Old McDonald's farm. So like horses, pigs, cows, sheep, rabbits, and all the rest of those. So all of these are mammals that can potentially serve as either primary or intermediate hosts for coronavirus, which makes it a little bit more challenging. So to bring this all kind of together and talk about why this is important for vaccine development, let's first start by just making sure we have a little bit of basic vocabulary for discussing vaccines. So just as a little overview for immunology, in immunology, we have two main sides to our immune system. We have what's called our innate immune system and our adaptive immune system. This innate system over here, which has a lot of your white blood cells, people are often familiar with things like neutrophils. This is like your first line defense. It typically is activated very, very soon after you have an encounter with a pathogen, sometimes within minutes to hours. But the innate immunity on its own, it sometimes can contain things and deal with things, but anytime the innate system is overwhelmed or it's evaded or it's bypassed altogether, we have to bring in the other side. So for the adaptive immune system, this is where we're going to get things like antibodies and T cells, if you're familiar with either of those. Now, these are extremely powerful and extremely useful and provide fantastic memory and long-lasting response. But one of the big problems with the system, if you look down here, it takes days to get this fully activated. To get even these T cells, it can be multiple days. To really get truly nicely functional antibodies, it can be up to 3 weeks before you get true antibodies. And we have entire chapters we cover in immunology about what is happening in that time frame that it takes 3 weeks to be able to get there. So this is problematic the first time you see something your body has never seen, because you don't have this really strong adaptive side that's going to be kicking in right away. And this is why people typically get sick. But the reason why people often don't get sick after they've been sick a first time with the same thing is because you store a memory in your adaptive immune system. So the next time you see something very specific, you kick this system on almost right away, and boom, it's in there to try to take care of immediately, typically before you even clinically feel symptoms. So this is the mechanism of how vaccines work. Vaccines are designed to trigger this adaptive immunity, create a memory response like you had been sick, and then be able to trigger it and bring it back out very quickly the next time around. So let's talk about antibodies for a minute. Antibodies are often represented as this little Y shape. They are proteins, and they like to bind to proteins, which is represented in this little illustration here. So importantly, an antigen is what an antibody likes to bind to. That's another one of those fun combinations of A and A. But more specifically, an antigen, which is something, anything that is foreign that your body recognizes, typically it's going to be a protein. Within an antigen, there are very specific sites that are referred to as an epitope. Here's what this looks like. As an example, here is hemagglutinin from influenza. Each one of these little Y shapes represents an antibody that's made against a different epitope on one entire antigen. This is important from a vaccine perspective because not every epitope produces the same level of protective response. Not every antibody is created equal, not every response is equal. So the challenge is getting a good response immunologically. We also are looking for something specifically called neutralizing antibodies, which are antibodies that specifically prevent the virus from binding to a surface and getting inside and releasing their genetic information. So you can have antibodies that can technically bind to something but don't neutralize, and those are not the same thing. So in producing a vaccine, the name of the game is producing neutralizing antibodies specifically. But T cells are also really critical, particularly in COVID. It's been shown that T cell responses in COVID are so powerful that you can actually create predictive models of whether patients are going to survive an infection based on how their T cells are operating. So the idea with the T cell here is you have two different classes. You have mild and severe disease. In a mild disease, T cells do what they're supposed to. You create strong armies of T cells and then strong memory cells that have a memory response. And then in severe disease, we have problems producing proper T cells. And there's a lot of questions about whether you produce significant memory cells to protect you again a second time around. Now, in terms of influenza vaccine development, what are we looking at here? Our main things we're looking at are surface proteins, hemagglutinin and neuraminidase. These are our primary antigens that we are developing vaccines against. And vaccines come in a variety of forms. These are some of our main types of vaccines. Our big one right here, our standard flu shot, is something called an inactivated vaccine, and it sometimes even comes over here to a subunit. What this means is you've taken a virus, you've totally inactivated it, so it's essentially dead. It wasn't really alive to begin with, but it's incapable of replicating in the body. You cannot get sick from it. And sometimes you even put it in a blender and then you just stick in pieces of it. And then the idea of this is that you have given the immune system enough to kind of grab a hold of it and create a response to it. So when people say that they get a flu shot and then they get the flu, technically, if you're getting an inactivated vaccine, you cannot. You can have an inflammatory response that makes you feel like you have symptoms, but you're not getting a virus. That only happens if you are getting what's called a live attenuated vaccine. So flu mist would be the example of this. This is where you take an active virus and you basically cripple it so that it goes into the body, but it cannot really replicate well. And this most closely mimics a natural infection. And there are certain people that are not supposed to get these vaccines, like the very elderly and immunocompromised, because there is a theoretical risk that they can actually come out and cause a disease. But most people get a standard flu shot, which is in this category here. Now, the big issue with flu and the big complaint on flu is that we have to get a new flu shot every year, and it's constantly changing. And here's the reason why this is problematic. We have to start producing influenza vaccine for a flu season in October for the US back in about February. So starting in February, they have panels that convene that take a stab at what they think is going to be the circulating strains of virus all the way down the road in October, and then they start manufacturing and producing them. So what inevitably tends to happen, since influenza likes to do this genetic drift, by the time we get to October, what's coming back around is often not what we originally thought. So we're trying to make a guess early on, and then we're kind of locked into a manufacturing production scheme. And then by the time we get to October, sometimes it's not super effective. And this is frustrating for people. And then it changes the next year, so then we have to do it all over again. So how do we fix this problem? Well, what scientists are trying to do is develop what's called a universal flu vaccine. So a universal flu vaccine, most importantly, would have what's called a conserved epitope, which means it's a specific thing that stimulates the immune system that is the same across all strains of influenza and is very stable, so it's not genetically changing, and it produces a good immune response. Seems like it would be an easy task, but we haven't gotten there yet because finding something that fits all of these criteria is challenging. But this is where we are currently, and this is where science is trying to go. How is this different from coronavirus? With coronavirus, our main target here is going to be our spike protein. In fact, there's two. We have the spike protein and then the nucleocapsid, which is another option, but most of the vaccines that we're seeing that are coming to market are focusing on creating a response specifically to the spike protein. The idea, if you have an antibody that can physically block that spike protein, it cannot bind to a host cell, it cannot get in, which means you cannot have disease. So that's how you were supposed to be protected by it. So this is where we are with our current vaccine landscape. There's actually this many different types of vaccines and this many types of classes that are currently going through clinical trials and testing phases. The two that I want to talk about today, though, are these ones down here, which I'm sure you've heard about. So the Pfizer vaccine and the Moderna vaccine. Both of these happen to be what's called a nucleic acid-based vaccine. What this is it's a messenger RNA vaccine. So we've seen messenger RNA here already. So Messenger RNA, its job is to produce a protein. It is translated into a protein. So essentially all this vaccine is you're taking this little piece of messenger RNA that gives the genetic code for the spike protein from coronavirus. And then you have to package it in a little vehicle to keep it safe, because messenger RNA likes to degrade really quickly, which is also why you have to keep it really cold. And so that's why they're having a lot of conversations about how we're going to be storing and shipping these, because we have to keep it cold to keep this messenger RNA from breaking down. So basically what you do is you take this messenger RNA. I know there's a lot going on here, but just to kind of explain what's happening, we take our messenger RNA, we bring it into a host cell, and then we translate it using the machinery into a protein. And then importantly, the protein gets put into these little things that are called MHC molecules on the surface of the cell. And you can think of these as immunological flags that are going to help to activate B cells and T cells to produce a response, to produce antibodies, and to produce memory responses. So instead of giving like an entire virus, you're basically telling your own cell to create the physical protein, because you've given it the instructions. It creates the protein and then shows it off and shows your immune system that it needs to create a response to that particular protein. So when it sees the real thing, it knows it's already seen it before, and you can create a response back. Now, what are the potential problems with this? The first and biggest problem is the fact that to date, there has not been a single mRNA vaccine that has been FDA approved in the US. Now, both Pfizer and Moderna were currently working on other clinical trials with other applications for these vaccines, which is one of the reasons they were able to develop this so quickly. They had a lot of the groundwork done that typically takes years to design and develop the vehicles and how it's going to work and interact. They already had that done. So then they just had to adapt it to the coronavirus. So it was very fortuitous. But the big issue here is the timing with all the clinical trials. So traditionally, this takes multiple years to be able to go through all the testing and safety and efficacy, and then also at the same time talk about the manufacturing scale-up process. What we're doing right here is we're condensing everything into a really short amount of time. Now, so far, these have shown good safety profiles, which is very encouraging, but what we don't have is any sort of data to show over long-term or in very high-risk populations what the potential side effects might be. What's also not discussed a lot in the news, which is true in the clinical trials for both Pfizer and Moderna, is that technically all participants had to get 2 doses, and it's what's called a prime and boost. So they had to get a first dose of vaccine, and then 21 days later, they had to get a second. So in terms of distribution and receiving vaccines, this is not if a vaccine comes out and you get 1 shot, you're fine and good to go like a week later. It takes up to a month to create that full response, and then you also have to get a second dose. So even as vaccines start coming out and rolling out into the new phases where the whole population might have access to them, it's not just a one thing and you're done kind of a situation. And then to finish with, the last big question that everybody's had is, will the vaccine be effective and last long term? One of the things that we do know from studying other coronaviruses historically is that the protective antibodies that are made in response to like the human coronavirus 229E, that's a naturally circulating one, it tends to fade. So for human coronavirus, every about 2 to 3 years, your antibodies drop off enough that you are no longer protected and you can technically be reinfected. We saw the same thing with SARS coronavirus, where we followed patients for years after they had been infected, and we saw that those antibody titers and the memory cells that make antibodies, that they die over time and we lose them. So what does this mean? It means so far we don't know what's going to happen with this because it's too early to tell, but there is a lot of discussion about needing to potentially have booster doses, potentially having this be something that's frequent, if not annually, then maybe every so often. And then we also are going to have to follow to see if things are changing genetically, that we might have to shift the vaccine around. So when we're talking about what long-term vaccination plans might look like in comparison to influenza, it may be more similar to what we see in influenza, where if we're not getting lasting protection, you may have to keep going back and getting other doses. But all of that remains to be seen because we just don't know. It's too early yet to know what's going to happen. All right, I think we covered quite a bit of ground. So I would be more than happy to take any questions relating to this or anything else on this topic. If you have a question that you want to ask that we either don't have time for or that you want to ask privately, you're welcome to send me an email, and I'd be happy to either schedule like a time to talk with you or I can email you back.
Okay. So there we are. Thank you again, Dr. Matthews. This is definitely an interesting topic and blew me away. So a lot of information there. I guess just to kick off some questions, I have one is kind of what are the differences between antibodies from actually having the COVID virus and getting a vaccine, and how does that differ?
That is a great question. So technically, from an immunological point of view, there is no difference. They are the same. And this is why vaccines are so powerful. So when you go through the process of creating an antibody, you have to actually kind of design it at a cell level. You move your genetics around and you change it up and you make it this nice little perfect antibody. It is the same antibody that is then produced later when you are exposed to a disease. So it doesn't matter if you get sick or if you get a vaccine, immunologically, you're producing the same kind of antibody.
And do those last essentially the same amount of time then? Is that because of the body, or does it
It
vary through the
depends.
population?
Yeah, that's a great question, and it depends. So one of the things that's frustrating with some vaccines and diseases is you can get some vaccines like, for example, smallpox vaccine is known to maintain antibodies for like 30 years. You can go in and still measure it and you still have them. Whereas you have somebody who gets sick with a coronavirus and they're gone 3 years later and you're like, what even gives? So the reason why those disappear is you have these memory cells that are kind of stored away. You can think of it as like putting long-term storage for like a computer. You set it aside. If you need it, you can pull it out and activate it really quickly. So we need those cells to be available to reactivate them when we get sick. But some vaccines do a better job of creating those memory cells than others. People typically have to go and get boosters for like tetanus. That's why if you go in like to the ER and you've had a wound, they ask you if you've had a tetanus vaccine in the last 10 years, because the type of vaccine for tetanus and the way it forms memory responses is not as strong or long lasting as something for like measles. That's a vaccine that produces a slightly different response. So this is one of the things we're gonna have to be watching. So when they say things like this vaccine is 90% effective, that's great. It's 90% effective at this point in time. But the big question is, how effective is it going to be as we go forward in time?
So that kind of follows up another question, I suppose, is right now we've heard a lot of this, a lot about this 90-day mark in terms of maybe having antibodies that long. Does it, and I'm trying to be very careful here because I'm not trying to advocate one way or the other, but if you get exposed to COVID again after already having it, does that maybe possibly extend the life of your antibodies then, or is that completely different?
It's another good question, and one we're going to have to look at. There is a concept in immunology that's known as boosting. And boosting can work both from getting natural exposure to an infection or a pathogen or an artificial one in a vaccine. So when you are exposed to, let's say you get a vaccine for coronavirus and then you're exposed to it. If you have gone through enough time that you have produced an appropriate response to coronavirus and then it comes in again, you're going to trigger everything in the system that it sees it. So then it's going to throw the whole system into like activation, and you're going to activate all these cells and they're going to go after that. And that actually helps to further solidify the memory response. So yes, like if you are vaccinated and then you're exposed to it later, even if you don't get sick, it may be helpful potentially for continuing to promote that memory. So we're going to have to see how that plays out, but that is a concept immunologically that does check out.
Thank you. So we do have one question on chat here. Why are some people symptomatic and others or I'm sorry, I think it should be asymptomatic and others get very sick?
Another great question. That is one that we are trying to figure out a little better. Some of it has to do with underlying medical conditions that have specific ties to the virus itself. For example, for individuals who are smokers, then you have higher ACE expression in the lungs. Individuals with heart complications can have higher ACE expression. People with kidney problems or kidney failure, if it's affecting the kidneys, you're going to have complications there. Individuals that have diabetes or severe obesity have a dysregulation in their immune responses. So when the immune system isn't coordinated properly, you don't get the right signaling for all the, essentially the soldiers to be in the right place to fight the infection. One of the big players that we're looking at in immunology is a particular cytokine, if you've heard this term. So a cytokine in immunology basically represents the signal that the system uses for communication. So it is a protein and it's sent out and it triggers responses in other cells. And there's one particular cytokine, it's called an interferon. And interferons, when they're produced in an appropriate window of time early in infection, do a fantastic job of getting your T cells up and ready to go and getting everything kind of lined up to help prevent infection. If that window isn't there with interferon signaling, or if it's too late, it can actually exacerbate the condition and cause a lot of complications. So there's many different hypotheses about potential genetic differences. There's the blood type difference one. But one of the things we've seen immunologically is that we think that this interferon is playing an important role, and different people may have different experiences with how their interferon is working. And we don't quite understand it, but so far that's come out as like the big player that we're looking at.
Great. Another one here, if you test positive for COVID and never have symptoms, what should you do?
Okay, well, there's two things that could have happened. One, if you test positive for COVID, you may have had COVID and you may have been asymptomatic. So if you test positive, the best thing to do is to self-quarantine and isolate, just to make sure that if you have it, because we have this concept of viral shedding, so even if you don't show symptoms, you could actually be spreading virus particles around in your environment. So we strongly recommend that you self-quarantine and isolate for the time frame that is recommended by like the CDC. The second option, though, is that there are sometimes false positives on these tests that come up. And so that's a little difficult thing, because unless you go and get a test to see if you have produced antibodies, you don't really know if you actually had it or you just had a false positive. So anytime you get a positive, just assume that you have it, and it's possible that you could be spreading it, and just kind of play it safe that way.
You can keep that on your if you want.
Can I mute this?
Yeah.
And yet,
I think we have some background noise here. Dose response, there's another one, dose response. Is there a threshold for amount of virus one can be exposed to, and more viruses, higher acuity?
Yes, this is an important concept in looking at transmission of infectious diseases. We have this, this idea called both the dose and the route of exposure, and that both of them can play a role in setting up how the fundamental immune response is going to happen. Say theoretically that you get a very low dose of virus that comes in through a route that's really not one of the preferred routes for the virus. So it technically gets into your system. So you remember, you have that first line defense of that innate system. A key of the innate system is that it doesn't kick in the next side until it gets bypassed or overwhelmed. So sometimes the innate system is able to handle things on its own, and it's able to kind of handle it, clear it, contain it, without having enough of an immune response that you clinically feel sick. This is important when it comes back to looking at this whole debate about masks, which has been a huge debate going on in our nation. One of the things that is a function of masks is, even though they are not 100% effective at fully blocking the spread of virus for a variety of reasons, what they can do is they can reduce the viral load that's transmitted out. So if you are starting with a lower viral load that you are getting, it can theoretically in a person's body make a difference between mild disease or severe disease, or even severe in death, depending on the situation for the person. So one of the reasons that public health officials have really been pushing hard on this mask and the request for people to wear masks, even though it's not a perfect solution, is it's about this risk reduction, and it's about reduction in the dosing of the viral load that's being eliminated, or reduction in the dose that you are receiving, so that as we're trying to control it as best we can at least maybe minimize some of the complications that are coming along with it.
Great. A couple more. Do you have, I know we're at the time limit now, but do you have a couple more minutes for a few questions?
Yeah, I'd be happy to stay for a few more minutes. People are welcome to go if they need to. And just for everyone who is here, thank you so much for participating. And again, if you have specific questions, feel free to reach out to me in email. I'm Matthews with 1T. You can also look me up on the website.
Thank you. And thank you everybody for joining. If you want to stick around for a couple more questions, feel free. If you need to jump off, we understand. Thank you for attending today's webinar. So the first one was, when a vaccine manufacturer says 90% effective, did everyone who have the vaccine get exposed to the virus afterwards? As an example, can they claim it worked 90% if they live in an area with lots of community spread?
Another good question. So one of the things that they're going to be doing in the clinical trials is they want to be able to quantify whether or not people were exposed to the virus. So that's a little harder to do, because typically you would measure that looking at antibody titers, but ideally you should have produced antibody titers from having the vaccine. So what they're going to also be doing is they're going to be picking people based on geographic regions where they can control some of those factors a little bit more to make sure that that's not potentially an effect that's going to be found in the study. So when they say that it is like 90% effective, what it means is that in their patients that came in that received this vaccine dose, they look at the number of people that then contracted coronavirus later after a follow-up in comparison to a group that received a placebo or something different. And they make the comparisons between the two groups to be able to see the differences. So you use what's called a control group as kind of your baseline. And if you have your groups that are very well in a well-designed clinical trial, when your two groups, your cases and your controls are very similar, you can kind of eliminate some of those what we call confounders. So things like if you live in a geographically different region, if you are of a different ethnicity or a gender, or if you are of a different age. So we match as many things as possible to eliminate anything else other than just the effect that we would see in the vaccine. So that's part of the clinical design process. So these really big companies like Pfizer and Moderna are very familiar with how to do clinical trials, and they have really good teams that create very scientifically robust clinical trials. Because one important thing is not all clinical trials are created equally. They're only as good as however well it was designed in the first place. But these are very, very big companies with a big track record for creating a lot of products using clinical trials. So I would trust that they have probably done their due diligence and tried to eliminate that as a factor in looking at an outcome.
Next question, if a person has had COVID-19 and has recovered, will the vaccine efficacy be negatively impacted?
So there is an interesting question about that comes up in the immunology literature, and it actually has to do more with if you've been infected with different types of coronaviruses before. The idea is if you have created an immunological response like to a different coronavirus, there are some versions of the vaccines that have come out where it's been questionable whether that interferes with your ability to create new antibodies. Now, some of that is going to depend on how similar the strains are for coronavirus, but expectations at this point in time would be that if you've already been sick with coronavirus and you get a vaccine, it should not affect that. You should be fine to go ahead and get a vaccine, and it should theoretically, hopefully, be more of like a boost. So like your prime was getting sick, and now your boosting event is going to be getting a vaccine to hopefully lock in some of that immunological memory. But some of those nuances, I know scientists are tracking it and they're studying it to make sure that that's not theoretically something that could come out. So somebody has an eye on that right now.
Great. Do things like vitamin D or zinc or anything play a role?
So technically, things like vitamin D do play a role in immunological responses. So it is an important component for overall immune responses. So a lot of people like to take it just to make sure that their immune system is functioning totally properly. There's not a specific mechanism to show that like taking vitamin D like blocks or interacts with influenza or coronavirus at a specific biological level. It's more about the idea of having the entire immunological system being kind of up and ready to go and respond at a moment's notice.
Well, great. Well, just had another one come in. What is the long-term effects?
The long-term effects of the coronavirus, the vaccine, what specifically?
It's a great response. I'm not sure. I suppose possibly of the virus itself.
So one of the big things that's come up from looking at patients that have recovered from coronavirus, even patients that have had mild illness, is there has been this whole big group of what we call, and has been referred to in the news and media as the long haulers. So these are individuals who have recovered from an acute infection, and now they are showing signs of more chronic illness. And a lot of the signs and symptoms that people are having are very similar to what people who have like autoimmune conditions or chronic medical conditions see and experience. So there's been things like, people talk about the severe exhaustion and severe brain fog, and sometimes there's complications that come with the heart. There's been some athletes who have been sick who have ended up having complications. So this right here is one of the reasons why, for example, for me as a parent, I have young kids and my kids are pretty healthy. And so we had a big debate about, do we send kids back to school? How does that look? You know, we have shown that children tend to be, you know, better at responding to the virus, but that doesn't mean that they are completely immune from not having these long-term complications. So just because you have had it and maybe you didn't have a lot of complications doesn't mean there couldn't be more serious things along the road. We do see in other viral illnesses that acute viral illness can lead to other long-term sequelae or complications that can exist. And it's yet again another reason why it's really nice if you can have a vaccine and prevent ever getting sick and having some of that damage that can be induced in the first place, you know, then it can protect against hopefully some of that long-term stuff that we're seeing. So I know that this is an area of emerging research. I would imagine in the coming years, we're going to be seeing a big influx of patients that if these things don't resolve, are going to be having to seek treatment for kind of chronic illnesses. And there's going to be kind of a big focus that's going to switch and start looking at mechanisms of understanding why is it when you're chronically sick that your brain gets really funky and you can't think clearly and you're really exhausted? Like, what are the mechanisms for that? So I think it's going to bring some renewed interest in exploring some of those areas a little bit more.
Wonderful. Well, again, thank you so much for doing this presentation today. And it was an amazing webinar, and thank you everybody for attending. So appreciate it, Dr. Matthews.
Thank you. I appreciate it very much.
All right. Everybody have a great day.
Bye.
That's a good day, sir.
Is she a doctor at the school?
Yes. I don't know.
Did she say that you can get it again?
Yes, that's all.